Hello friends, welcome. Myself, Amol Vidate, Assistant Professor, Department of Plant Pathology, Sadhguru College of Agriculture, Mirasgaon. Today we discuss about diseases of the sugarcane, which are the major crop growing in our area. That is red rot of sugarcane, smut, that is whip smut, then rust disease, red strip disease, sugarcane mosaic, ratoon stunting, and set rot or the pineapple disease of the sugarcane. In that first one disease, that is red rot of sugarcane, which is caused by Coletrichum falcatum at perfect stage, that is Phycelospora tachomenensis. So here is the photograph. We clearly seen that is symptoms of the red rot, that is typical shortening of the internode and red color strips which is appears on the leaf surface. Symptom, that is first external symptom appears most on third and fourth leaf, which wither away at the tip and along the margin. Typical symptom of the red rots is observed in the internodes of the stall by splitting it longitudinally. These include reddening of the internal tissues, which are usually elongated at right angle to the long apex of the long axis of the stall. The presence of the crosswise and patches are the important diagnostic characteristics of the disease. Pathogen. The pathogen are disease cycle. The fungus is a seed borne and also persists in the soil on the disease clumps and stubbles as chlamydospores and dormant mycelium. The primary infection is mainly from the infected seeds and secondary spread in the field through the tools, irrigation water, and cultivation practices. The rain splash, air currents, few dewdrops also help in the spread of the conidia from disease to the healthy plants in the field. Management adopt crop rotation by including rice and green manure crops. Select the seeds from the disease free field, disease free areas. Avoid ratooning of the disease crop. Soak the seeds in a 0.1% carbendazim or tridamophoam solution for 15 minutes. Then, before sowing, grow resistant varieties like Co 62 198, Co 7704, and moderately resistant varieties like Co 8001, Co 8201. Seeds can be treated with aerated steam at 52 degrees Celsius for 4 to 5 hours by moist air treatment at 54 hours for 2 minutes for 2 hours. Next disease, that is whip smut of the sugar cane, which is caused by Ustilagocytamine. For the symptom, it produces the calaminous symptoms of smut. The affected plants are stunted and central shoots are converted into a long whip-like dusty black structure. The length of the whip varies from few inches to the several feet. In early stages, this structure is covered by thin, white, papery membrane. The whip may be straight and slightly curved. The pathogen, the fungal hyphae, are the primarily intercellular and collect as a dense mass between the vascular bundles of the host cells. Produces teeny black spores. These thin membranes which covers the smut weep represent the host epidermis. Horrible condition, monoculturing of the sugarcane, continuous ratooning and dry weather during tillering stage. Disease cycle, teleospores may survive in the soil for a long period up to 10 years. The spores and sporidia are also present in the infected plant material in the soil. The smut spores and dormant mycelium also present in or on the infected sets. The primary spread of the disease is through disease sets. In addition, sporidia and spores present in the soils also spread through rain and irrigation water and causes soil-borne infection. Management, plant healthy sets taken from the disease-free area, remove and destroy the smutted clumps, Collect the weaves and thick cloth bags in polythene or the plastic bag and immerse in the boiling water for the 1 hour and kill all the spores. Discourage the rattoning for diseased crops having more than 10% infection. Follow crop rotation with a green manure crop and dry fallowing. Then grow the red gram as a companion crop between two rows of the sugar cane. Grow resistant varieties like Co 7704, moderately resistant varieties like a Co Change 86061. Third one disease, that is pineapple disease of the sugar cane or set rot, which is caused by Cerasistus paradoxa. The disease primarily affects the seeds usually 2 to 3 weeks after planting. The fungus is a soil borne and enters through the cut ends and proliferates rapidly in the parent camatose tissues. The affected tissues first develop a reddish color and turn a brownish black in the laterally stages. Pathogen, the fungus produces both macroconidia and microconidia. Conidiophores are linear, thin wall with a short sail at the base and the long terminal sail. Microconidia are hyaline when young but become almost black at maturity. They are thin walled, cylindrical, and produces endogenously in a chains in a long sail. Favorable condition for the disease causing poor drain field, heavy clay soils, temperature 25 to 30 degrees Celsius, prolonged rainfall after planting. Disease cycle Disease cycle, fungal survives as a conidia and chlamydospore in the soil and in the infected buried canes in the tissues. The inoculum moves from field to field through the wind-borne conidia and irrigation and rainwater. The seeds are spread rapidly through the parenchymatous tissue. Management, soak the seeds at 0.1% carbendazim solution for 15 minutes. Use long seeds having 3 to 4 buds. Provide adequate drainage during rainy seasons. Wilt, that is Cephalosporium sacheri, the first symptom of the disease is visible in the canes of 4 to 5 months age. The canes may wither in a groups. The affected plants are stunted with a yellowing and withering of the crown leaves. The midrib of all the leaves in a crown generally turn yellow, while the leaf lamina remain green. The leaves dry up and stem develop hollowness in the core. The pathogen, the fungal mycelium, is hyaline, septate, and thin wall. The conidiophores are simple or branched and produce single celled and hyaline oval and elliptical microconidia. Favorable condition, high day temperature, low humidity, low soil moisture and alkaline soil, excessive dose of the nitrogenous fertilizer. Disease cycle, fungus is a soil borne, remain in the soil as a saprophyte for 2 to 3 years. The disease is primarily transmitted through infected seeds, secondary spread by the wind, rain, and the irrigation water. Management, select the seed material or seeds from the disease free plots, avoiding the practice of the returning in the disease fields. Burn the trashes and stubbles in the field. Grow coriander or mustard as companion crop in the early stage of the crop. Dip the seeds in the 40 ppm boron or manganese for 10 minutes or 0.25% imisan or 0.1% carbendazim for 15 minutes. Fifth one disease, that is rust, which is caused by Buccinia erianthe or melanocephala, that is typically bloody red pustules which develops on the upper lip surface. Symptoms, minute elongated yellow spots, uredia, usually appears on both the surface of the young leaves. The pustules are brown on maturity. Late in the season, dark brown to black tilia appear at the lower surface of the leaves. The pathogens, the mycelium is hyaline, branched and septate, produces ovoid and pear-shaped, single-celled uridospores measuring apical thickening and the golden yellowing color. Favorable condition, it required 30 degrees Celsius temperature, relative humidity 70 to 90 degrees Celsius, high wind velocity and continuous cloudiness is favors for the disease development. Then disease cycle, the fungus survive on the collateral host like Erianthus fulvis and uridospores also survive in the infected stubbles in the soil. The disease through airborne secondary spread by the airborne uridospore management, removal of the collateral host, spread tridamorph 1 kg or the mancozeb 1 kg per hectare. 6-1 disease, that is red strip, which is caused by Pseudomonas rivulinians. Symptoms, the disease first makes its appearance at the basal part of the young leaves. The strips appear as water soaked long narrow chlorotic streaks and become reddish brown in a few days. Pathogen, the bacteria is a short rod gram negative non capsulated with a polar flagellum. Favorable condition, continuous retaining and prolonged rainy weather with a low temperature 25 degrees Celsius. Disease cycle, pathogen remain viable in the soil and the infected plant residues. The bacterium is also survive in the sorghum, pearl millet, maize, finger millet and other species of the saccharum. The bacterium primary spread through the infected canes and secondary spread mainly through the rain splash, irrigation water and insects. Management, whenever the disease is noticed, affected plant should be removed and burned. Growing resistant varieties, selected seeds from the healthy fields. Avoid growing collateral host near the sugarcane fields. Seventh one disease, that is sugarcane mosaic, that is mosaic potty virus, sugarcane mosaic potty virus. The symptom, the disease appears more prominently on the basal portion of the younger foliage as chlorotic and yellow strips alternate with a normal green portion of the leaf. As the infection becomes severe, the yellow strips appears on the leaf sheath and stalk. The pathogen, it is potty virus, it is viral disease cycle, which is transmitted by the infected seeds and by the maize and the cereal, that is sorghum vulgare. Management, the use of the resistant varieties, roguing the infected plants and disease free planting material. Then uh, that is removal of the maize near the main crop. Then last one, that is rotund stunting, that is Clavibacter xylem, subspecies xylem, rickettsia like organism, that is RLO. The symptom, disease clumps usually display stunted growth, reduced tillering, thin stalk and shortening internode, yellowish foliage, orange red vascular bundles in the shades of the yellow at the nodes are seen in the infected canes. The pathogen, that is Clavibacter xylem, is a areolo known as presently in the xylem cell of the infected plants. They are small, thin rod shaped corniform, 0.15 to 0.32 micrometer wide and gram positive. The primary spread of the disease through the disease seeds. The disease also spreads through the implements contaminated with the juice of the disease canes. Maize, sorghum, southern grass, cynodont serve as a collateral host for the pathogen. Management, select the seeds from the disease free area or from commercial nursery. Remove and burn the clumps showing the disease incidence. Treat the seeds before planting and specified for grassy shoot disease. Thank you. Please like, share my YouTube channel and subscribe my YouTube channel.